Okay, so in the channel here, I gave the list of today's agenda. What we are going to do first is to revisit this fate algorithm because we didn't have enough time in the lecture to explain each step in this fate. And I will describe some features of fate which different from the t-SNE and UMAP. And then how can we use that feature to do the trajectory analysis? I'll show you some examples. Then we will spend the time to study these built-in functions, k-means, db-scan, and the spectral clustering algorithms. Those are the built-in functions provided by MATLAB in the statistic toolbox. So it's very convenient for us to use this built-in function. Just call that function directly. And then we will visit the SC3 algorithm. Let me edit this a little bit. We will look at the source code and see how each step, the source code implementation matched with the paper described steps in the PowerPoint slide. Remember, we have that. This, okay? So we will look at the source code and see each steps in the source code which match to this pipeline here. Then I will play a video. This is a real application from scratch of the after embedding, then to the clustering, then to the annotation of cell. And this is a talk given by Michael Salinas. He's a postdoc in nutrition department. When we had a meeting and we recorded his talk, and it's very good examples as a real world applications of this pipeline. Which pipeline is this whole thing, okay? This pipeline. And the last, we should still have time, and we'll see how we can install R, because there are a lot of programs, packages for single cell data analysis has been developed, implemented in R. We don't have to reinvent the wheel for each case, so we can see if this R package is very useful for us, how can we, from the MATLAB, from the interface of our cell population, and I call that R function, and compute over there, and get back the output from R, and transfer back to our interface. Show you some examples in there. Okay, so in MATLAB, I'm going to do the update. You know, I can use this command line to download the latest version of the package and unzip. If you don't have this, you can Google to see the quick installation. I want you to spend the time to update it because there are some updates recently if you haven't got time to do. So quick installation, if you search the toolbox, because this procedure takes some time. basically what I typed in the command line is this particular line of code in this quick installation. Okay, if you do that, we can let the package to be updated. Well, in the meantime, we are going to revisit this page. So this page is the pipeline of fate. This is a program used for reduce dimension of the high dimensional single-cell RNA-seq data. It starts from the matrix of the gene expression. So this is the UMI matrix. Then you compute a distance between each cell. So each point is a cell. So you can do this, use PCA, reduce dimension, then compute the distance. It's also possible to directly compute the distance between cells. Then as long as you have a distance matrix, so this will be a big matrix. If you have 2,000 cells, the matrix will be 2,000 by 2,000. Each position tells you how far from one cell to another. Then the next steps are all the different kinds of transformation, filtering, enhancement to make this distance matrix better capture the global structure and also reflect the distance between cells. So that's idea is about consider there's a manifold. Okay, we will talk about this high dimensional mathematical structure later. And anyway, so this transformation allow you to obtain a better, more informational distance. Then from there, you can do a MDS to get this trajectory, to get this embedding position of the cell. So in this illustration, this structure is not much different from this. Okay, the input here and the final outcome. But in reality, if you have PCA embedding results, you will see, okay, this is very different. You can hardly to tell the structure of data. PCA will give you not so distinct description of the cell population relationship. But at the end, you get this better one. Let's come back to here. So let's see. I do have this complete. If I do single cell, so if you look at this function, so this function is located in the package under a folder. If I open this subfolder of this package, this function is embedded into a subfolder called +run. So inside this, I have subfunction, the function which inside this folder, somewhere in here, okay? I can call this function using a syntax look like this. Put a run dot, so the program will be able to, MATLAB will be able to find that function which inside the subfolder there. So this gives you a basically this domain name, so you can have a better organization of your function in there. So I put all the additional function which you run either using MATLAB implementation or R implementation inside this run subfolder. Okay, so that's how you organize the source code. If you look at this open, so this is a function of the fate. So what's the input of fate? This function takes big X, that's your expression matrix, Gn by cells. Then you ask for a number of dimension of your output. Okay, the S is your output that's embedding the position of your cell after the computing, after your computer gave each cell position in the 3D or 2D. So you use this number of dimension to decide whether you want output to be 2D or 3D. So the input will be X. The input is X, remember? So you have number of genes, gene 1, 2, gene M, and that's your C, C1, C2, Cn. So that's your input dimension. So you put in the fate program. Your output is number of C. Okay. C1, C2, Cn. There's a number of cells. So for each cell, you have either 2 numbers, 1, 2, or 3 numbers. Why? So this matrix called S, I use S as embedding. So its embedding is from this high dimensional, so that's M dimension. To 3D or 2D. So you reduce M dimension from here to 3D, and this is called embedding, where that's embedded, so reduced dimension. From here, I can do a scatter plot because I have 3 numbers for each cell. So this X, Y, Z. So this will be X, Y, Z. 3 numbers coordinate. So each cell, based on the 3 numbers, I put in this space. It will show up the result of embedding. Is that clear? So it's the same as a t-SNE. You can replace this with a t-SNE, UMAP. They're all the same. Same kind of dimension reduction, okay? Yeah, I think what we can do is we can use a real data set. We can load example data and let to run this to see how we can get S, then to plot them. So I can load example let's see where I am. I can load, let's see. So I'm inside. So don't have to type. You just put a few characters, then you put a tab, should be able to find this. So we have this object called SCE, and then we extract from SCE object the X matrix. So I just prepare a preload example data, which is X. Yeah, it depends on where your current folder is, and this may change, but as long as you can point to the right example data, you should be able to load it. Mm-hmm. To play with, if you look at the size of this, and so M is the number of rows, so you have 7,173, 37 genes and 2,000 75 cells. So that's the dimension of X. Now, we want to get S out, so run MATLAB underscore fit. Yeah. So this is just get the dimension of X. You don't need this if you can see that here, so you have an idea how big this is. So I want to just give you some impression what should expect from the output of this. So now you gave the three, let's see 2D. If you ask for a 2D output, and this embedding S will become only 2 columns. Okay. We are calling the fit algorithm to run. You will see some feedback, the output from the program. It shows the intermediate steps. You will see PCA, you will see the computing of the matrix distance, and at the end you will see the MDS. So it gave you it's doing PCA before compute the distance. You see at the end doing this MDS. The diffusion, the landmark chain computing, it used spectrum clustering in the middle somewhere. Okay, it's done. Look at our output S. S contains 2075 rows and 2 columns because we ask for first 2 dimension embedding. If you do this, put a 3, you should get 3 columns of embedding. You can double click and to look at this numbers in the 3 columns. So that's your X, and this is your Y, this is your Z, X, Y, Z. So you put the cells based on their relationship, also considering global relationship between each cell's distance to another cells. So everything considered. And then project this back to the 3D space, keep relationship between them because of the algorithm we are using. Now we can plot those points, 2075 points, using the scatter plot. Scatter 3, so because I'm using 3D, if you don't have a 3D, you only have 2D, you can use this scatter. Scatter plot, this we will use the coordinate of the first one here. So that's my X, my second. Remember, I pick the second column and the third column, X, Y, Z. Okay. Let's see what you get. We should have a very similar figure. Not exactly, but almost the same, right?
Yeah.
Okay. Good. Okay. Good. Okay. Yep. So according to our schedule, next we are going to do the k-means to do the cluster. For example, I want to cluster this as a group, right? And then this cluster, maybe, so depends on the algorithm we use. So the input for our clustering algorithm mostly will be this 3D coordinates. So we are going to use embedding. We cluster cells based on their 3 numbers each, rather than using several thousands of the dimension. Because this program have already help us to group the cells with similarity, high similarity into small, into the neighbor. So give our good foundation to do the clustering analysis. So we can start from here rather than from the very beginning. If you start from here, you'll still be able to work because you can do k-means using so many parameters to define each. But the k-means, when it compute the distance between cells, it will not deal with the distance so nicely as a dimensionality reduction. The algorithm was specifically designed to look at the distance between there and the transformation and to make this clear group. If you do the k-means directly from here, you rely on the estimation of a distance, which you know when the dimension is really high, you don't have very good estimation using the original data. But rather, if you use the distance between cells using this, because this has been processed already, so give you the better results in general. So we are going to do k-means with this, okay? So use this as input. K-means, put S as input, and the k-means will give us the index, basically, ID. So if you have a cell belongs to the first group, it will give you 1, another first group 1. So this is the first group. Then if you have another group, you will have 2, 2, 2. Basically, it's just an index for each cell. And here we have our example is 2,075 cells. So the output of k-means will be a long index. How long this index is? It's 2,075 numbers, just 1, 2, 3. And if you have a cell number 1, see, it's a group 2, so you have number 2. The second cell is group 3. So you have this. You have these numbers. And using these numbers, those index, you know this cell and this cell, these 3 are cluster number 1. And this and this and that is cluster number 2. Those are index. Okay, so what do we need to have a k-means like this program to run? We need the coordinates, need the data, and we need a hyperparameter, which is a number k. So we need to give k-means a number. So from here, I see, okay, we can give 5, 7 maybe. Just give it 7. So we are going to do the 7 k-means to get 7 output groups. And in the index, you will have a number from 1 to up to 7, because you ask for 7 groups. So that's the function. So I will give IDX, the index, k-means as a let's see, seven. This should work. I'm not so sure. Let's try it. It's done. So if you finish this, you get a new variable called index, IDX. How long this IDX? 2075. You can double click and see. The largest number in this index should be 7, and the smallest should be 1. So this is okay. Now, we can use this index to color the cells based on their membership. For example, I want to color all the cells in group 1 using red, and this using blue, and those using green, something like that. So I can achieve this by using the scatter plot 3 and then add the index somewhere. Let's see. So this position this position gives the size of your this, and the index will have you group. I hope it's working. Now you have different colors in your plot. If you count, you should have 7 different colors, and each color indicates a cluster. as I said, we can use, so let me change it back to 7. 7 looks like all right for me. So as I said, you can use this to do embedding, okay? Just make sure you directly embed the original matrix. That will take longer. And also make sure the rows, the rows are the cells and the columns are the variable define the property of each cell. So you have to transpose this to give this. So I just want to see if we use not embedding, but use the original matrix, what we can get. So this result looks not as good as we cluster directly using the embedding. You see some of these groups. This may be cluster number 1 occupied quite a lot of cells. And those cells dispersed into several small groups. Well, there's a mixture of this in here. So the reason is this extremely high dimension. There's some of the structure, so you cannot have a very good estimation of the real distance from one cell to another. Just like I said, the manifold, if you look very close in the high dimensional space, but in reality you have to follow the trajectory, move all the way along the objects of this complex. In that case, the estimation of used in the k-means to estimate the distance between cells, not so accurate. That's why I gave you the results, which looks not as good as we expected. Okay, so in this case, we will keep using the embedding to do the clustering. Okay, you get this. Now, you can try dB scan. If you do the dB scan, you see, okay, this is a function in the toolbox built-in function. So I gave you a job to use this function to do embedding, to do the clustering, to replace k-means. What you can do is to look at the documentation of this function and then to get yourself familiar with this and see whether you can work out a solution for using this function to do embedding. Give it a try. The output will be very similar to this. You want to get the index of the membership of the clustering number. But you replace this k-means using dB scan function. and then read documentation and see how you put the other variable, the input variables in order to make this program run. So in this case, you still take this, give the embedding, the coordinates of cells as the input for the program for this algorithm. But you need two parameters. Remember, the parameter define how long the radius is, how many minimum numbers of cells to be considered in that region. The circle is defined as a, so you can call a cell a core or border or outlier or something. So you need to play with the parameters here, the two numbers, to get the results and see which number give you better results. So this function, if you look at the documentation, this is since R 2019A. Compared to many other functions, this is a relatively new function. Has been introduced into MATLAB since the release of 2019 version. So if you use the previous version, say 2018, you don't have this implementation. This is a relatively new function, dbScan, to do spatial clustering with application with noise. You need two parameters. One is epsilon, and another the minimum points. In our input is not X, not the original, but it's using embedding to do this, okay? If you look at the example here, this is the example data used as a test. So you have scatter plot in 2D, but the points are distributed. Looks like this. So dbScan takes that positions, X, and using 1 and 5 as two parameters. So we probably can try 1 and 5 and see what we get. So the output is the same. It's index. There's -1. I assume -1 just indicated outliers. So let's try scatter, put out those numbers. And we should try other numbers. So this is how long the radius. Okay. So maybe we should change this to make this longer, because we have coordinates from 0 to the range 250. So we have much larger range. So maybe make this as 5, and also this make it the like 6. Okay, let's see what we get. Okay, looks more reasonable. If you look at your index, you have 1, 2, 3, and we have a few -1. So I'm pretty sure -1 are the outliers to be considered by this algorithm. So I try maybe another number, 7, and here put 8. Let's see what we get. Okay. Okay. So we need some of these combinations of the two parameters, because it change the results greatly. So if you put smaller numbers, you may not be able to capture enough core points, and then you put a lot of it as outlier. So this is one of the limitations of a DB scan, although it's very useful when you have the shape like this, and then you have some idea about the range. So you put 1 for the distance, and you give the good results. Well, if you do the k-means, you get something like this. K-means based on the distance. Okay, so the last one you should try is which spectral cluster. So if you look at this function in the toolbox of statistics, and you can try to read the documentation. And please follow the documentation and try to learn how to use this, and then try onto this data set here. Okay, let me know once you get the result. So it's time for the break, 5 minutes, okay? Okay, so from this documentation, we can see this is also a relatively new function introduced in 2019. You need 2 parameters to make this run. The first is the data you put in. You can use either the big X, or in our case, we use the S using embedding rather than X. And you need a K. So this K is a number of clusters, okay? Similar as a K-means. So the way we can do is to do this spectral cluster with S and 7. And the outcome looks like this. Okay, spectrum clustering, the last step is also K-means. If you read through the pipeline of the clustering algorithm, we had that in the literature here. So this spectrum clustering algorithm, and you start with a adjacency matrix, which is a network, and then compute the degree, compute the Laplacian matrix, then eigenvalue, eigendecomposition, and from here, you take this and to do the K-means, basically. Okay, so that's why this algorithm requires to have a number K, so how many clusters you eventually want to get. Then the algorithm not work on this data directly, not like a K-means, the native K-means algorithm, but it do some transformation through the network of those cells. So that's why you get something look like this, okay? So in our pipelines, we not using DB scan in the toolbox I created. We will just use K-means directly because it's most straightforward to give you the best results. Interpretable because you have the embedding, you know the relationship you get. Just group them into cluster, not a big deal. That's why I did not include DB scan in the toolbox. I did not include spectrum cluster. Rather, I just directly use the K-means to get this separations of the cell populations based on the embedding. Okay, now I'm going to play a video which, before we talk about SC3. SC3 will be the algorithm that surpass all these embedding steps, directly work on that to do transformation, to do extraction of information, then K-means, and put multiple K-means together, directly on this. But before we do that, I will play the Michael's video and to let you have an idea. So once we finish this embedding and the cluster, for example, you use the K-means, give you the results, and what you can do in the real application. He basically illustrated the steps after cluster and the annotation of cells. He worked in the nutrition department, so his sample is from the clonal sites. from a mouse in testing. So I'm going to play and see how the should be okay.
So it's really the identification of all the different cell types in the mouse colonic tissue. So there are some data processing steps. The first one is filtering based on the QC plot, and I'll talk a little bit more about that and how I decide what to filter. Then it comes to the identification or the separation of colonocytes versus the non-colonocytes. These non-colonocytes are really just going to be the immune and the stromal cells that are also in these samples. And then once I can separate those, I can focus on the colonocytes, and I can then start to try to identify all the different subtypes of colonocytes in these crypts. And I'll talk again more about this in a bit. So this is the colon tissue. So we isolate this colon tissue. This is a human, but we get it from mice. And we have now started using the whole intact tissue. And what I mean by that is before we were just kind of taking off this top part, this epithelial layer, and then sequencing that. But we do know that down here is in the muscle layer is where all the blood flow is, all the immune cells are, and they are also responding to things like diet. So we're now interested in looking at the whole colon tissue. So we now have all tissue there. This is kind of what it looks like at a cross section. At the top here, you have the epithelial cells. These are the colonic crypts that are right here. And then down here, this is where you have your immune cells, your blood vessel cells, muscles, all different kinds of fibroblasts that give nutrients to the colonic crypt stem cells at the bottom. It's all there. So everything is important, and I think it's good that we have started doing this, but it does make the data a little bit more challenging. And as you can see within these crypts, there are a ton of cells really squeezed in here. And even in the center of the crypt, some of these circular things are part of goblet cells. So if we use a different staining, we can kind of see that the blue here are those goblet cells. And so we can capture all of this with our single cell data. So we have all of these different cell types here in the colon. This is just a simplified version of that. And this is what we've been using to do some RNA-seq. So the first thing, like I mentioned, is the QC filtering. This is really going to depend on whether you're using Flex or the 3' sequencing. The Flex is really clean data. You almost don't need to filter this data. If you do filter it, it would be just to kind of get rid of these cells that have a high mitochondrial percentage. But everything else here, to me, kind of looks fine. But if we come over here and we look at the 3' expression data, if we just look at the number of genes, this graph here, just from my experience, I've learned that down here in this section is really kind of junk cells. So we want to kind of filter this out to where it looks a little bit more like this over here. Even in the number of reads here, where there's this break right here, below that is kind of just these junk cells that, again, that we need to filter out before we begin processing the data. And that's how I filter my data. I have to look at the QC plots and kind of just go from there. Again, the Flex data that I'll be showing today is pretty clean, so I actually didn't perform any filtering on this data. This data comes from gnotobiotic mice, that is mice that don't have a microbiome. So this is pretty much just pure colon tissue, unaffected by the microbiome. This is what the data looks like when we load it into Dr. Sai's software, and then we can begin to embed it. I like using the UMAP because it just makes a lot more sense to me biologically compared to the other embedding methods that you can use. So the first thing when we have this plot, like I mentioned, is going to be trying to separate those colonocytes from the non-colonocytes. I use two major markers for this. I use EpCAM and ECadherin. These are going to be expressed highly in epithelial cells. And this is what I use for me to separate the colonocytes, which are EpCAM positive, from the non-colonocyte cells over here. Of course, this isn't enough. I have to go in and really dig deep into what these cell types are. So just so that you guys know, about 60% it's really about 50 to 66% are colonocytes, and the other percentage, 34%, is going to be these non-colonocytes. And again, this is going to vary. These are mice that don't have a microbiome. So in the normal wild-type mice that we do see, this is going to change a little bit. There might be more immune cells because of the microbiome. I'll just really briefly talk about the non-colonocytes, but it won't be my main focus because these cells are actually pretty easy to identify. You can see that they make up just these little clusters here, and they separate really nicely, and we can identify them. And we get all different sorts of immune cells, B cells, macrophages. Sometimes I see dendritic cells, sometimes not. There's also blood vessel cells, so endothelial cells, smooth muscle cells, mural cells, and pericytes that are all connected to the blood vessels. Then there's all sorts of fibroblasts, some myofibroblasts, and then some other types of fibroblasts that, like I mentioned, are providing growth factors and nutrients to the colonic stem cells. And then we also see things like neurons and even adipocytes show up in this data. This is just a heat map for all of these cell types. So they do kind of break out really nicely. And I'm pretty confident in these cell types too, but I haven't put a lot of focus into this. I haven't really analyzed data coming from the non-colonocytes just yet, because my major focus has been on the colonocytes. So this is when I subsetted out the colonocytes, and then I begin to try to cluster these cells and see what are these cell types. So I use k-means clustering, and I kind of just play around with the numbers until I find one that really separates out everything kind of the way I like it, like visually, and then I can kind of go from there. So the first thing I do with the colonic cell types is going to be separating out the enteroendocrine cells and the tough cells, and that's just because they actually stick out from the data. So in this case here, this cluster 11, I know is going to be the enteroendocrine cells, and this cluster over here, number 3, is going to be the tough cells. And I know that by the marker genes that they're expressing as well, and we can actually calculate a score for these marker genes for the enteroendocrine cells. So you can see here they scored really highly compared to the rest of the clusters. That's cluster 11 up here, so I know those are enteroendocrine cells. And the tough cells, again, scored really highly and different from the rest of the cells. That's number 3 right here. So these guys are tough cells. That's easy. That's actually pretty easy and shouldn't be too much.
It's hard to me, but
like again, it's just because they actually pull away from the rest of the cells, because they are a differentiated cell type, but they are technically a specialized cell type as well.
Expression is so different; they just separate.
Yeah, exactly. The other cell types that I can try to pull out are goblet cells. Goblet cells are going to be this whole chunk right here. And I know that based on the MUC2 expression, so you can see that it expressed very highly in these clusters here. So I know those are goblet cells. I can also calculate a score for them, and they do separate out compared to the rest of the clusters down here. So I know, again, these are goblet cells right here. So I can actually subset out the goblet cells again, and then begin to focus on the specific goblet cell subtypes. I know nobody really does this, but I thought it was very interesting because once I saw this, I remembered a paper that I had read that actually mentioned that there's 2 lineages from the goblet cells. There's a canonical and a non-canonical lineage, and that's actually what I observed here, so that was pretty cool. Down here, what I'm showing is MKI67. This is a proliferation marker, so you can see that the clusters about right here are going to be the cells that are proliferating.
so that's interesting because they're supposed to be generally quiescent, right?
Yes and no.
So there are actually some cycling cells in there.
Yeah.
They're going to divide.
There's also differentiation markers. So for goblet cells, a good differentiation marker is MXD1. And you can kind of see that, I didn't do a good job of lining these up right, but down here are going to be where those proliferating goblet cells are. And actually this little cluster right here that you see is secretory progenitor cells, or what I think are secretory progenitor cells. And then we have the non-canonical goblet cells here using these 2 markers that I grabbed from that paper. So this kind of branch right here is non-canonical goblet cells, and these over here are canonical. You can kind of see that this non-canonical branch is actually very close. That's this branch right here. It's very close to the other colonocytes right here. So it was very interesting to actually see that out on this 3D map. But based on those markers,
That's a biology.
yeah,
That's
To
right.
us, just two branches. We don't know.
yeah, I know, and I
We needed to read it.
I remember that paper. So for the secretory progenitor cells, they have different names and people call them different things. That's what makes this also very complicated. So I started calling them secretory progenitor cells. I believe previously we were calling them deep crypt secretory cells. So they do have this Reg4 marker. You can see that cluster number 12 is scoring really high for this Reg4 marker. That's this cluster right here. And you can also see that clusters 10 and 12 are also proliferating. This is a proliferation marker, so I know these are proliferating right here. DLL1 and DLL4 are expressed again in these secretory progenitors, and you can see that cluster number 12 is scoring high. It's actually the only one that's kind of scoring. So to me, these are secretory progenitor cells. This is what it looks like once you actually apply those cell types. So we have the secretory progenitor cells here. We have some proliferating goblet cells here, and then the rest, even though there's two lineages, I merged them just to simplify things. Those are the differentiated goblet cells. And actually, if we look at the differentiation potency, you can see that the secretory progenitor cells score really high, and the proliferating goblet cells, and then the differentiated goblet cells at the bottom. So this looks good. That's what I expect from this data. Like I mentioned, we have these differentiated cells, but they're also very specialized cells. They have nice functions. And so you just saw that I was able to identify all these cell types here. The next thing would be to identify the stem cells and the absorptive colonocytes. So you can kind of see from this image to this image, I got rid of all those specialized cells. And now we're really left with the backbone of what I guess colonocytes do. So the colon is largely responsible for absorbing any sort of water that the small intestine didn't absorb. So that's what these cells, that's their function, and that's why they're called absorptive colonocytes. And so what I did was I subsetted out the stem cells and the absorptive colonocytes. They form kind of like this gradient, very similar, I guess, to this crypt right here. They form this gradient from stem cells and transient amplifying cells down here all the way up to differentiated cells over here. So I know that because of LGR5 expression is located down here at these clusters at the bottom. You can see that these are also the same cells that are proliferating based on proliferation markers. And then there's some differentiation markers that I use. I like to use KRT20 to identify the absorptive colonocytes. And aquaporin 8, this is, of course, a receptor that is responsible for pulling in water into the cells. And that is what I use to identify the late absorptive colonocytes. I don't believe I actually have an image of that, but this is what I'm currently working on right now. This is what I've had to do to be able to identify these cell clusters. My initial thinking when I first started all this single cell stuff was that, we'd be able to have our cell atlas with all our gene markers and cell types and just kind of, click and play and it'll identify stuff for us. I learned over a year or two that is not possible at the time, especially not for this particular cell type. It works actually pretty well for things that separate out really nicely, like blood cells, like peripheral mononuclear blood cells. Those do separate really nicely into T cells, B cells, NK cells, and I can actually identify them using a cell atlas. But for the colon, I just haven't been able to do that. So this is what I have to do. I just have to look at the expression across these figures here and then try to label these clusters here as a specific cell type. Again, this isn't finished just yet. I need to input a lot more details into this so that somebody can actually follow it. But for the most part, this is how we identify cell types in the colon.
So that's why, so you have potentially another approach that you were trying to optimize using the larger genes. You described it before a little bit. Is that something you're still working on, more general?
For which one? For this location of cell type?
Yeah. Was Victoria doing that? And you were taking I call it the personalized approach versus
data driving.
So Salim's osmosis, active trend. First question you want to ask is, he has a mouse that has no microflora in a strange state. but it is. The thing is, also just do a very quick, and then he has manipulations where they added one bug and a substrate, or plus or minus a substrate for a bug, and maybe nothing changes from the Atlas standpoint. Really, because it's going to be doing something very interesting. Oh, they'll have the other information as well that could be added. So I think that this is wonderful.
Okay, so this is Dr. Michael Salinas. He did this analysis a year ago using the toolbox and focused on the subclusters. So we separated the cells into many different small clusters and deal with maybe one cluster each time, or consider several clusters as a whole subtype of cells. Nowadays, we try to make the whole procedure fully automatic. But indeed, we do need to have a better understanding of the system you're working on. In this case, Michael is familiar with the system. He knows what kind of cell type, subtype of cells he expects to see, and with markers, he's familiar with those markers. It's really hard to replace human annotation with using this automatic procedure. We try to make this ChatGPT-like stuff to automate everything, and there's something we are working on actively. But still, this is like a typical procedure. I hope you can learn from what Michael is talking about, how you are going to deal with individual clusters of cell. For this. Okay. So you have this video in the chat channel, so feel free to revisit it when you have a real data set, you want to know the steps. Okay, now let's see IC3, the algorithm. I cleaned the desktop, but not the variables, because we are going to use this X to run IC3. This is MATLAB implementation, SC3. So it will return a variable called C. C should be the index. That's CLS, what the CLS is. Okay, so C should be the index, and your input is the big X matrix. Clustering algorithm directly from the expression matrix. So we don't have to go through to get embedding of the cell and give S to the cluster. No, we will do this directly. Then the next parameter you need to have is the K. K is number of the cluster you want to get. So let's just write this out. This put X here, and this put ML underscore capital S C3. Okay, so this is how you use the algorithm. So you can see the reference, and I have the URL to the article. Yeah. Yeah, so this is in the toolbox. So you have to have the toolbox in the. You don't have this. Uh-huh. Mm-hmm. Yeah. Do you have the toolbox in your pass? You have. Mm-hmm. Looks like you put a 2 underscore. Yeah. Oh, there's a barcode. So maybe we cannot try the MATLAB version to get the same problem. Yeah. So I can quick check those what the message is about. Yeah, so I do found a problem here in this step here. So basically, this is the last step to put all the output from the previous k-means algorithm and then to assemble the results. So this rely on the function of this. So there's something wrong with this function inside here. So it's another function inside here. Yeah, so this is maybe it's a bug, maybe it's in this situation we logic. So we cannot test this at this moment. I have to figure out what's wrong with this particular problem. And I will update you later. So this happened inside one of the sub-function which assemble the cluster. I think I can show the source code to let you know each steps. We have a bug in the last step, but the previous steps are okay. So I take this X matrix in. So the first step is to do the normalization and the log transform. Then use a function to get the distance matrix. So if you look at this, step 1 is to compute a Euclidean distance matrix. That's between cells. If you come back to the pipeline of this paper, it is this step to compute the distance the one we are doing is compute the Euclidean distance matrix, the step we are using. Okay. So this is a Euclidean distance matrix. Then start to compute the distance using Spearman correlation. So this is a correlation function. Compute the pairwise Spearman correlation coefficient between two cells. If these two cells have all the genes co-express, then the correlation is large. Then 1 minus it will be the distance. Because if you have a strong correlation, then the distance between you will be small. So this is the second step. The third step is to use Pearson correlation distance. Okay, you just change the same function to compute the correlation coefficient from the S Spearman to the Pearson. So this 1, 2, 3 steps are these three. Okay. So for each of these Spearman, each of this distance matrix, it will go downwards to PCA transformation, Laplacian transformation. So those downstream steps is inside. So once you get this distance matrix, then we will go to this function. Okay. So this function, if I search for this function, inside this function, we will do PCA. We will do the Laplacian transformation. So for each distance matrix inside here, we do two different transformations, which are this PCA and Laplacian. Okay. So for this transformed matrix, it starts to do the D-range, basically sliding using different range combinations. That's where these steps are. Okay. So for the different ranges, and each time extract different ranges from the transformed matrix, and then to do the k-means. So this k-means inside the D range are those. Okay. So the output of the D k-means will be put together. So this is the concatenated, all these outputs, and make this as like a cluster index array. So this cluster index array, once finished, will be sent back by this function. So this cluster array sent back, and then they will be put together because we have three different matrices of a distance. The last step is to ensemble them. So that's where the bug comes from. The last step we have a problem to get this. But that's the very last step. I'll let you know once I fix this bug, we can try this next week. Okay? So that's our SCE. Up to here. Okay, now let's stop here, and also I'm going to clean my desktop and clear all. I can let my working folder back to the original here. So let's just load an example data from this interface. Let me delete one file. So in the import data, you select the example data. Then let's see, processed data. So this is a processed data set. And also it's fully automatic because this is a very simple data set. If you have a case in your own study, maybe you need to follow the Dr. Michael Salinas procedure to annotate this. What I want to show you is the manual here is some external function which some of this labeled with R in front of this. So those are the R package you want to call. For example, you want to detect ambient RNA contamination using R package called decontx. This R package, the methodology is published in a PubMed paper. You can find this original paper by using the PubMed ID. For example, you want to run this, you cannot just directly click here because your R environment is not installed yet. So we need to know how to install R package. So if you click, there's no R package has been installed, so we need to find a way to set up. In my case, I already found the R interpreter here, so I can select. Yeah, so I need to install this package, Surite maybe. So depends on your, something wrong with my installation. So there's no package, for example, has been set up. So we needed to set up this R environment. So what you can do on your computer is to visit this page, ZenWorks. Then from here, let's see if R Studio. You should have this icon in your ZenWorks, and we should be able to open and install. Basically, it will update the R environment on your machine, make it ready. Then we can link to this newly installed R environment from the MATLAB, so we can use the package in the drop-down menu over there. The which one? From the MATLAB? Are you using Mac? No, Windows? Yeah. Yeah. Yes, maybe something wrong with the link. Yeah, sometimes the first time maybe not run, but maybe second time, because the first time it's try to install the package. Yeah, it's quite a lot of stuff. It's not easy. So so this is a communication between two languages. Basically, you have a X matrix from MATLAB right into R and get back information. So there are a lot of potential problems in between, but we try to streamline this procedure as much as we can. So we need to have this installed and then make sure from the R Studio just installed, you can have from here, so the R Studio, you can have this R interface. The same thing, you have to know where you installed your R. So R Studio say, okay, let's detect machine default. Yeah, so for this one, we are using this version R4.32. Let's just install a package from here, installed, for example, Surwrite. So we need Surwrite, and then that's where R is working. So once you have a Surwrite package installed there, the package you run from the MATLAB should work because you have that already installed in the R. Otherwise, it takes some round to install it automatically, maybe. So remember, this is some kind of advanced applications. We are dealing with two different languages. You should have it here. Yeah, double click. Double click. Yeah, R Studio. Yeah. That's right. Okay. It worked? Detected by the package, yeah. Yeah, you have R Studio. Okay, maybe. So you see in my newly installed R package, the Surwrite rely on a lot of others, so it just keep running. So once it's finished, I should be able to come back to the MATLAB. This is just put in the background, but if you want to use R, it's okay. You can run from there. So I can just put this as a back. I come back to this interface. Now, in the external, I need to reset the R running environment, because now I point to the 4.42. This is not the correct version. So I want to find C programs are, so I have this version is correct, so I should be able to put the right position. So now I set environment right, and then come back here. So I want to run Surat workflow. I just installed it, so you should be able to run it now. See, 2D. Which one? So this one I'm trying the Surat package to let Surat to process the data for me. Yeah, it doesn't matter, whichever you want. Yeah. So you can see this is the R is running. Then I come back. So Surat package using UMap, I believe that we will come back at the UMap embedding. Looks similar to what we can run using UMap here. Yeah, I don't know what happened. Yeah. But yeah, it's very slow.
But it completes it, and it says installation is complete. And then when I go to click on it, it does the same thing again.
Yeah, you just click once.
Oh.
Yeah, you don't
Double click.
it. Yeah, it's once it's finished, it's finished.
Okay.
Yeah. Yeah, so you have to go to the binary and then select this. Yeah, then you set it up. Yeah. Now you can try external, then maybe the detection of this. Yeah, anything. It's okay. Yeah, I think it's okay. The first time maybe is installing the package, then you run that again after the first round. The Surat load. Oh, package was built. Yes, something. Yeah, so I don't know the latest version of R, maybe the 4.4 something.
What's the problem with this one? But this one is 4.32.
Well, I don't. Yeah, I don't know if you. Yeah, I don't know how to solve this R problem. Yeah. Yeah, I don't know. Yes, yeah. If it's your own computer, you can install the latest version. But now this computer only allows you to install old version. So there's some problem with it. Yeah, I think it's fine. It's already. You don't double click.
Oh, okay.
Yeah, it's already installed. So from here you can have the R Studio. So this already there.
Oh, okay.
Yeah. So you can shut down this. Yeah. Yeah, so if you cannot solve the problem today, it's fine. So even in R, I have some problem, though, because this indicates the R version we installed is different from the Surat package it's supposed to have. So we just cannot run this because R is not correctly configured out. So I have problem to run this because this incompatibility of the R package and the Surat package, R version. So I have to solve outside here in order to I can move forward. So this is, so maybe I should try inside here to try the contamination remove. So this will be another R package. So the first time it will install that package. If you look at the output, after installation, you run that again, this should work. Now is install the package. I think that's all for this week. See you next Tuesday. We will discuss classification and some neural network. So, okay? Mm-hmm. You still.
So I got the R stuff to load.
Yeah.
So now it says matter connecting it back to matter, right?
Yeah.
So I can.
You can, yeah, start from here and then load. You don't even have to load. You can set up the environment by set up R environment. Yeah. And then see, yes. Basically, you try to find where this is located. Yeah, click this.
This one.
Yeah, and you are done. Then you should be able to run. Okay.
Thank you.
See you next week.
It's okay if this one's not fully done. Oh, this is done. Okay.
Mm-hmm.
Okay.
Mm-hmm. Yeah.